Hello, this is Albrecht Koppenhöfer. I am the Extension Specialist for Turfgrass Entomology at Rutgers University. I will here present to you findings of our ongoing research on sampling methods for the annual bluegrass weevil, one of the major insect pests of golf courses in North America. So, the objectives generally are to optimize the use and the predictive power of the existing sampling methods for ABW adults. In particular, we're looking at the effect of height of cut, HOC, and temperature on adult detection in mower clippings, in vacuum samples, and in soap flushes. Moreover, we're looking at the effect of water volume and detergent concentration on the adult detection efficacy with soap flushes. Now, for the HOC effect on adult detection for both clippings and vacuuming, we used mixed poa annua creeping bentgrass areas on our hort farm 2 in New Brunswick. We used areas with a height of cut of 0.375 inches, so lower end fairway, and of 0.118 inches, so representing a green. Into each replicate of each plot in each field, we released 40 color-marked adults into the plots about 1 hour before the extraction to give them some time to get comfy and distribute naturally. After the actual mowing and vacuum, which I will explain later, the plots were then soap flushed with 0.4% lemon scented liquid dishwashing detergent solution with 2 applications after 0 and after 5 minutes of 500 ml, so 1 pint per square foot area. And when we observed adults and collected them for 20 minutes to determine the total adult population. Then the adults were collected and counted, and then at least for the mowing, we examined them for survival and injury under a dissecting microscope in the lab. So for height of cut effects on adult detection in clippings in particular, we used plots that measured 6 by 2 feet. The clippings were collected into the baskets of a Toro Flex 21-inch mower, and the baskets had attached to them, or not, a brush. You can see down there in the picture that was supposed to, not only brush the grass, but somehow then also pick up more adults. We'll see if that worked. And so here you see, on the left side in the graph, you see the data from the fairway without and with brush, and what you can see out of the 40 adults that were released, we only recovered next to nothing on the fairway. We recovered a lot more, but still not that many, up to 5 adults on average on the green with the brush. Then here we see the data with the soap flushes, so we see that the soap flush discovered a whole lot more additional adults after we had done the mowing. And what we also see is that there were about twice as many adults recovered from the fairway, and that we explain that probably in the shorter mowing height the adults could just disperse more quickly, and a lot of them might have actually left the plots. And then finally, to get us a better idea of what really happened, relatively speaking, we look here at the percentage of the adults that recovered in the clippings relative to the total recovery, so including the soap extraction. And then what you see is that on the greens, we recovered between 15 and 24%, with the brush 24%, so that was a decent percentage of the adults of the total recovery. So that could be a useful method, but on the fairway that was hopeless, so you can't use the clippings to find adults on the fairway. That would be completely useless. But you have to be very aware that a lot of the adults, in fact 85% in our experiments, irrespective of green or fairway, survived the mowing, and 70% actually were undamaged. So they could just go right back to business. So obviously you need to discard these clippings if they have significant numbers of adults in them far away from any playing surfaces at risk or even destroy them. So, for height of cut effect on adult detection using vacuuming, we used an Echo ES250 model leaf blower in which we inverted the airflow, and then we inserted these mesh baskets, you can see that in the lower left corner, to capture any adults and as well also debris that was sucked up by the vacuuming. And then we vacuumed the entire plot. Each plot measured 2 by 2 feet, either none times or 1 or 2 times for 10 seconds each. So you can see here in the bottom the actual vacuum and the inlet, and then you can see me here vacuum up an area, and then on the lower right side you can see my 2 postdocs going through a container with the clippings to see and count the adults. So what we found is that if we just look at what we found in the vacuums, in the picked up material, that there were about 3 times as many adults picked up from the green as from the fairway. Not terribly surprising, they're just more exposed there. What we also see is that neither on fairway nor on the green did we pick up any significant additional number of adults by a second pass of vacuuming. Then here you see the total numbers that were then picked up by vacuuming as well as by soaping afterwards. And once more you see that about twice as many adults were recovered total on the fairway than on the green, which is again probably an indication of that the adults on the greens just could disperse more quickly, and a lot of them might have dispersed outside of the plots. So then, to give us a better idea of a relative recovery, here in the percentage you see in the columns is the percentage of adults recovered by vacuum relative to the total number, and you can see that in the fairway that is about 4 to 5%, which is not a whole lot, but you can use that to get an idea of what's going on. Whereas on the greens it's 29 to 33%, and so that's a pretty good recovery rate, and gives you a good idea what's going on. Then finally, we looked at how to optimize the soap flush method. For here, for this experiment, we used natural ABW infestations. We used our lemon-scented liquid dishwashing detergent prepared in solutions of water at 0.2, 0.4, and 0.8%, which is equivalent to a quarter, half, and 1 fluid ounce of detergent per gallon of water. Then over each 1 square foot plot, we spread out 500 or 1000 ml, which is about 1 pint or 2 pints. We did that either just at the beginning of the observation period or in the beginning and after 5 minutes. And then we collected the adults that were flushed up to the surface in 5-minute increments up to 20 minutes. So here you can see then the effect of the concentration. You can ignore these different colors. They're just how many adults were found in the different time periods, 5-minute intervals. But you can see here, as you go from left to right, with increasing percentage, the number of adults increased, and the highest recovery was found with 0.8%. Here you can see the effect of volume, so you can compare the effect of how many were recovered with 2 applications of 16 fluid ounces pint, 0.4% versus 1 application only in the beginning. You see that about twice as many adults were picked up with the 2 applications. You can also compare here again the effect of water volume applied once, so 1 application of 16 ounces recovered only about half as many as 1 application of 32 ounces. So overall, if you look at the data, it seems like 2 applications of 16 might be somewhat better than 1 application of 32, but the difference is not statistically significant. Also, with the 1 application of 32, you either have to do that extremely slowly, or a lot of the material is going to run off. So it's really not very feasible to apply that much material at once. So then in total, you can see that overall, the 2 times applications at 0 and 5 minutes of 16 fluid ounces at 0.8% detergent was the most effective and feasible way. And if you look at the time intervals now, you really needed to collect the adults at least for 15, better 20 minutes to really get a good representation of what's going on. So, in summary, overall we saw that clippings only work on the greens, 15% were recovered there, and was hopeless, 0.2% in the fairways. Vacuuming was more effective, 29% on the greens than on the fairways, where we only recovered 4%. But 4% you can still work with. It's, people use this method all the time. Then on the greens in particular, vacuuming was more effective with 29% recovery than going through the clippings, which recovered 15% without the brush. And as far as soap flushes go, they were more effective than vacuuming. For example, on the fairway, we recovered about 8 times as many adults through soap flushes than vacuum. And on the greens, even on the greens, we recovered 3 times as many adults through soap flushes than through vacuuming. And finally, just overall, soap flushes clearly by far were most effective because they recovered really the majority of adults with at least 75% of the adults. Now, in ongoing studies that we're doing and will continue to next year, we're looking at the effect of temperature on these different methods, and so far, I can really only say that, not surprisingly, the detection efficacy goes up with temperature, and so somewhere below 60%, the adults are just not very active, and it becomes less and less effective. You recover less and less adults, and we expect the same with the vacuuming. The soap probably is going to be much less, but we don't know yet. So, if you want to look for some more information on this, look at my entomology webpage at Rutgers, where you can find extension presentations and publications. And if you have any other questions, you can email me or give me a call. And finally, this research is funded by the U.S. Golf Association and by the Center for Turfgrass Science at Rutgers University.